PharmaVentures, experts in deals and alliances.
Hello and welcome to PharmaVentures Insights here in Barcelona. There are many in silico companies all telling us that they have a great way of predicting efficacy and safety for drugs and shaving time and capability off how drugs are developed. The proof of this is yet to come to reality. But one company's actually been doing this for quite a while now and licensing programs out to pharmaceutical partners. And they're going a little bit further than that. They're actually taking their own technology and developing drugs with it. With me today is Josep Prous of the Prous Institute, who's going to tell us a little bit more about his company and how they're using in silico technology to bring about the next generation of drugs in both CNS and oncology. Josep, welcome to the show.
Many thanks.
So the Prous Institute, it's not a name I'm familiar with. You're a Spanish company, I guess. Perhaps you could tell us a little bit about what it is you're doing and the technology you're bringing to the healthcare sector.
Yeah. The idea of the company was to go one step further compared to the provision of factual information and offer new predictive analytics and data mining technologies to identify the efficacy profile of drugs and the safety profile of drugs in front of thousands of targets. So this in silico approach permits to rationalize and to optimize the need for experimental validation, which is needed, of course, at all points in time in biomedical research. So this is a short summary of what we've been doing. The technology has been implemented in a variety of pharma companies who utilize these tools to investigate new drugs. Also adopted by public research organizations and regulatory agencies, probably more on the safety side of the story. And finally, we have been using this technology to initiate our own drug discovery programs.
So as you say, you're going one step beyond, and the technology that underpins it has already been validated by Big Pharma. So they're using it already.
That's it.
So now you're taking that back internally and using it to develop your own drugs as well. Are you focusing on any particular therapeutic areas?
Yeah, we have developed a variety of programs. One is cancer. We have been investigating what we call autophagy modulators. Autophagy is the process by which the cell cleans itself of unwanted residues and process products. And by modulating neuroplasticity, we are able to interact with diseases like cancer, CNS. This research program was licensed out to a pharma company. And we continued developing other areas. One was in the field of diabetes with SGLT2 inhibitors. And the one which is very active now is in the field of CNS disorders. So we have been studying what we call neuroplasticity modulators. Neuroplasticity is defined as the way the brain reorganizes itself based on internal and external stimuli. And it has been observed that in a variety of CNS disorders, ranging from Alzheimer's to autism and others, neuroplasticity has a big impact. So what we want to do with our molecules, having applied the technology on top of a variety of targets, to identify molecules that can bring neuroplasticity processes back to normal.
How far do you take are you planning to take the development process? You've got your in silico model, which you know works. You say you're doing the wet biology side of it. How far will you go?
Yeah, so the idea is to move the projects up to IND stage, so to perform the preclinical toxicology package, and then license out the molecule to probably a pharmaceutical partner. This is what makes more sense. We have been doing a variety of experiments for these neuroplasticity modulators. In particular, a very attractive one is the availability of a Fragile X model of disease, a knockout model, because these reproduce quite well the abnormalities which are seen in cerebral tissue in Fragile X post-mortem. With the administration of the compounds, we have been able to see how we are able to restore the dendritic spine density and maturity back to normality, which is a very important physiological biomarker for these findings. We have also tested the molecules in other paradigm, cognition, visuospatial memory, so a series of protocols which try to reproduce what is seen in patients.
So given your company's been doing what it's been doing for a little while now, are there actual drugs that have made it to market yet that have been based upon your original technology?
We know that some of the molecules derived from our first autophagy program are in clinical trials,
Okay.
so a second generation of these molecules. And for the ones in CNS, we look forward to start the clinical trials soon in 2018. So we need to go through the preclinical toxicology package and eventually start probably in a neurodevelopmental disorder like Fragile X syndrome. And provided that neuroplasticity is affecting other diseases, we may move this to other pathologies as well.
So pretty soon we'll know how good this is, and hopefully the answer is going to be
Hopefully,
it's
yeah.
very good.
Hopefully.
Josep, thank you very much for joining us today.
Thank you. Pleasure, Adrian. Thanks.
For more information about PharmaVentures, visit our website